Okay, welcome back, everybody. I think everybody's here. Um, the next speaker is Sarah Tishkoff. Sarah is the David and Lynn Silton University Professor in Genetics and Biology, also here at Penn. She's the director of the Penn Center for Global Genomics and Health Equity in the Department of Genetics. She studies genomic and phenotypic variation in ethnically diverse Africans, combining fieldwork, laboratory research, and computational methods. Her talk today is entitled Global Patterns of Genetic Variation and Association with Clinical Phenotypes at Genes Involved in SARS-CoV Infection. So why don't you go ahead, Sarah, and share your screen.
Yep, just about to do that. Let me try one more time. That works. Not working. On one moment, let's try it again. Have it on this. And share screen. Not showing it.
So we were able to see it briefly the last time you did it.
Okay, let me try it then one more time.
Okay.
I find that often you have to click right on the PowerPoint before it shows up. Well, thanks to Susan and Rick for inviting me to present today on behalf of a number of collaborators who are shown here. And I just want to highlight a few people who played a key role in the project that I'm going to present. And that includes Chao Zhang, Huanchen Feng, and Mike McQuillan from my lab, Marcello Mello, I hope I'm saying that right, from Cesar de la Fuente's lab, and Anurag Verma and Giorgio Sarugo. But before I present that work, I want to start by talking about why I think it's so important to include ethnically diverse populations in human genetic studies. We need to do a much better job characterizing both genomic and phenotypic variation in populations from across the globe, and to understand the evolutionary processes that generate and maintain that variation, to understand gene-gene, gene-protein, and gene-environment interactions and how they contribute both to normal variation and disease risk. The focus of my research is on Africa. There are a number of reasons for that, but one of which is the fact that all modern humans originated in Africa. These red dots represent the locations of fossils of anatomically modern humans, the oldest of which is dated to around 300,000 years ago. So after this, around 50 to 80,000 years ago, a relatively small number of people migrated out of Africa, giving rise to populations across the globe. So this demographic history and adaptation to very different environments and very different pathogens has shaped the pattern of genomic variation that we see in populations today. The other thing that motivates my research is to understand risk factors for health disparities. So for example, hypertension is more common in people who self-identify as African American rather compared to other ethnic groups in the US. Type 2 diabetes is more common in Native Americans compared to other ethnic groups. And of course, we see dramatic health disparities in regards to COVID-19. I pulled this off the CDC website last night, showing that there are relatively more deaths per 100,000 individuals among people who self-identify as African American or Hispanic compared to other ethnic groups. Now, we know that there are major socio-demographic factors that are contributing to these health disparities, things like also unequal access to healthcare and probably systemic racism. But it's thought there may be genetic risk factors as well, and certainly, almost certainly, there are going to be individual genetic risk factors that influence why some people get severely ill from COVID and some people don't. And yet, as we showed in this paper we published a couple of years ago, there's a major European bias in human genetic studies. We looked at the Genome-Wide Association Study database and showed that it was composed predominantly of Europeans. They made up about 80% of the individuals in studies. Around 10% were Asian, 2% African, 1% Hispanic, and less than 1% everyone else. And we see a similar European bias if we look at other common databases like Nomad and GTEx, this is really problematic. It impedes our ability to fully understand the genetic risk factors for human disease, and it could exacerbate health inequalities. Our ability to translate genetic research into clinical practice or public health policy could be incomplete, or even worse, it could be mistaken. It could result in inaccurate assessment of pathogenic variants in clinical genomic studies. And one of the reasons for that is that one of the criteria for determining if a variant is pathogenic is how common it is, with the idea being if it's very common, then it's less likely to be pathogenic. Well, maybe it's not common in Europeans, but maybe it's common in other ethnic groups, which would change the classification of that variant. And then lastly, attempts to use estimates of genetic risk, for example, polygenic risk scores from European-based studies in non-Europeans could result in inaccurate assessment of risk and lack of appropriate intervention. So for that reason, my lab and our African collaborators shown here have been doing fieldwork in Africa for more than 20 years now, and I thought I'd show you a bit of what that's like. So we're focused on studying populations that are living more traditional lifestyles. They tend to live in very rural areas requiring use of a four-wheel drive vehicle to get there, and we have to bring all of our supplies with us. Here in Tanzania, you could see how we're doing some of our biometric analyses. Here are some of the researchers in Botswana and showing some of the tremendous amount of ethnic diversity that we see. Here are collaborators from the University of Yaoundé Medical School and members of my lab who've made important contributions to these studies. But because we're really concerned about doing this research in an ethical manner, the first thing we do, in addition to getting IRB approval at Penn, is that we have to go through very rigorous ethical review in each country. We have to get a permit to do the research, but well before we're in the field, we're having community discussions. We translate into the local language. We talk about the risks or the benefits of any of the research and any concerns they have, ultimately getting community consent and then individual consent. We think it's really important to return results to participants. I think that's a way to, a version of benefit sharing. And training and capacity building in Africa is really important. I've had the honor of training a number of grad students and postdocs from Africa, and the hope is that they will go back to Africa and lead the research there. So we're in these populations, we're obtaining blood, and from that we can get DNA and RNA and plasma. We get detailed ethnographic information about diet and any medical history they have. And then we have the challenge of processing these samples with typically no electricity. So as shown in the lower left, we've started bringing our generator with us, so we can just set up the lab in the bush. And then my lab is using an integrative omics approach, integrating these different types of omics data together with information about environmental factors and how those are influencing normal variable traits and disease risk. So what I'm going to tell you about today is a study of the pattern of variation at genes that play a role in SARS-CoV-2 infection. Specifically, we've studied ACE2, TMPRSS2, DDP4, and LY6E. We're interested in understanding the evolutionary forces that impact the pattern of variation. So why should we even care about that? Well, we know that there's an arms race that's going on between the virus and the human host. The virus is constantly evolving, getting new mutations that enable it to resist the human host immunity. The human host is also adapting and evolving. And so if we look for signatures of natural selection in the human genome, we might be able to hone in on functionally important variants that are common because they're important in playing a role in infectious disease resistance. Today, because of a lack of time, I'm just going to focus on two of these genes. So as you know, ACE2 is the receptor for SARS-CoV-2 that it uses to enter into the human host cell. TMPRSS2 is very important because it's going to cleave the ACE2 receptor and activate the spike protein, which is going to facilitate the entry of SARS-CoV-2 into the cell. But ACE2 plays some other really important roles. It's also a key regulator of blood pressure and renal function as well. So the data set that we use is a novel data set consisting of whole genome sequence data that was generated as part of the TopMed consortium at NHLBI. We were looking at a subset of the data, 2,500 whole genome sequences from more than 70 highly diverse ethnic groups. And here in the upper right, these different colors represent inferred genetic ancestry. And you can see that there's a lot of variation within and between groups. So it's representing a tremendous amount of diversity. And then we compared this data to the 1,000 genomes data set, which includes global populations. We looked at the coding region and the intron, both introns and coding region and regulatory regions as well for each gene. So starting at ACE2, this is on chromosome X. It's 127,000 base pairs long. The majority of the variation that we find a lot of novel variation in these diverse Africans. Most of it is in non-coding regions and predicted to be regulatory variants. But we do see a number of non-synonymous variants. And in fact, we identified 41 non-synonymous variants. Using computer software programs, we predict that many of these variants are predicted to be potentially functional, have an impact on protein function. Now, we used a statistical test of neutrality which looks at the ratio of non-synonymous to synonymous changes at a gene in the coding region of a gene. And under neutrality, we expect that value to be 1. If it's very negative, it means that that gene is under purifying selection. It's highly conserved. And if it's greater than 1, it means that there's diversifying selection. So HLA is a classic example where there's selection for lots of diversity. What we see at ACE2 is purifying selection. We see a lot of rare non-synonymous variants, with the exception of East Asia, which has an excess of non-synonymous variation and no synonymous variants, which is interesting. But the only populations that we see common non-synonymous variants are in two groups called the Baka and the Badjeli, who are so-called pygmy populations from Central Africa. And I'm going to come back to that in just a moment. First, I want to tell you about work that was done by Cesar de la Fuente's group. They looked at the impact of these non-synonymous variants on protein structure. And 2 of these variants are novel, non-synonymous variants, are found directly in the RBD binding region so could influence the binding with ACE2. And this other variant down here occurs in a region identified as the TMPRSS2 cleavage site in ACE2. So it could potentially interfere with TMPRSS2 cleavage efficiency. And then there are some variants over here which are impacting glycosylation. We don't know yet what the functional impact of these are. That there needs to be few additional studies to determine that. So going back to those 3 non-synonymous variants that we found that are common only in pygmy populations, I will mention that one of them is in LD with a regulatory variant nearby. And as shown here, these variants are really only found in pygmies. Now, the pygmies eat a lot of bushmeat, and they have a high exposure to animal viruses and documented relative resistance to viruses. So we just found it intriguing because it makes us wonder, could this reflect some kind of selection for resistance to these types of viruses in that population? So these non-synonymous variants and the one regulatory variant that's linked with this non-synonymous variant are on 3 distinct haplotype backgrounds. So that's how the different alleles are arranged along the chromosome. And we wanted to ask the question, are these common in pygmies because they're under selection, or is it just by chance that they got this common? So we use something called an extended haplotype homozygosity test. And it works like this. Imagine that in red, this is a new mutation. And that mutation increases the fitness of the person who has it. So they have more children and their children have more children and so on. It's going to rise to high frequency in the population. and it's going to drag with it the neighboring variations. So if somebody's homozygous for this red variant, they're homozygous for the yellow variant and the blue variant, and for many variants, going out hundreds of thousands of nucleotides. And that's exactly what we see here. So down here at the bottom are the homozygosity on chromosomes that had the ancestral alleles, and we see it's not very far. But on the chromosomes that have the non-synonymous mutations, we see extended haplotype homozygosity over hundreds of thousands of base pairs, suggesting that it may be under positive selection in that population. The other thing that we found that is, I think, really interesting are a number of regulatory variants that are common in global populations. But there's the set of 6 that were particularly interesting. They all pretty much overlap this very strong DNase hypersensitive site. This is located about 73,000 base pairs upstream of ACE2, and CHIA-PET data indicates that it's interacting with ACE2. And in addition, it is an EQTL in brain and some other cell types. So those 6 variants are in LD to some extent with each other, and they form 6 haplotypes. And we could see that outside of Africa, there's just these 2 haplotypes. And interestingly, the haplotype shown in red is all the alleles on that haplotype are associated with decreased expression of ACE2, at least in some of the cell types for which we have data. And in East Asia, they have just the haplotype, predominantly the haplotype shown in blue. In Africa, we see this haplotype shown in yellow. It's most common in hunter-gatherer populations, the Pygmies and the San from Botswana. And then in this purple color, we see another haplotype that's only found in the Pygmy. So those are a potential future interest to look at functional studies in the future. We then extended this test of homozygosity across the whole region. We used a statistic called IHS. And if this dot is above the red line, we highlight it in red, it's significant. Or if it's below the red line, it's significant. And the point is that we see a lot of variants that are targets of positive selection flanking ACE2. These are going up to about 200,000 base pairs away, a cluster of predicted regulatory variants, as shown here. And some of these are over this very strong DNase hypersensitive site. They are shown to interact with ACE2, and again, they are EQTLs in certain cell types. Now, these potential targets of selection are in very strong linkage disequilibrium. This is showing pairwise linkage disequilibrium in global populations. So it's hard to know what is the functional variant. There may actually be a number of functional variants, but I think that there is clearly selection for gene regulation, for gene expression regulation in this region. And so I was excited when I ran across this paper that was published by Regeneron and together with a number of collaborators here at Penn using data from the Penn Biobank. They did a genome-wide association study with 52,000 COVID cases and about 700,000 controls. And as other studies have shown, they found a strong association on chromosome 3. There's a number of genes in that region that are potential candidates. And they also found an association with the ABO locus. But they also found an association with ACE2 on the X chromosome. And interestingly, they found that there's a rare variant near ACE2 that lowers gene expression and protects against COVID-19, and it's variable in frequency in different populations. So I think this is something that should definitely be followed up on in the future. I'm just going to briefly mention some of the results from TMPRSS2. Again, the majority, we find a lot of novel variation. Most of it is non-coding and predicted to be regulatory. But we do find 48 non-synonymous variants. Most of these are rare, so we don't see any evidence for diversifying selection. And in this case, there are a couple of these variants where we see that they're very common in assorted global populations. I'll just highlight a couple of variants that we found in this predicted regulatory site that's very close to the promoter of TMPRSS2. And what was interesting was the global frequency distribution. So here we can see that the minor allele shown in red is at almost exactly the same frequency in every population. And I must say that's very odd. I don't typically see that. When I see that, it makes me think that there might be balancing selection, maintaining those alleles at a particular frequency. But the only exception is East Asia, where the major allele shown in blue is at almost 100% frequency. So this is a potential interesting variant to follow up on. So we applied another test of neutrality called the mcdonald-kreitman test. I want to tell you a little bit about this. What we do is we compare the sequences in humans to the chimp genome sequence. And we are thought to have diverged from chimps somewhere between, say, 5 to 7 million years ago. They're our closest genetic relative. So we're counting up the number of variants that are fixed, meaning that all humans have them, that are non-synonymous differences from the chimp, and we compare them to the number of synonymous fixed differences from the chimp. And we also look at the ratio of non-synonymous variants polymorphic in humans and synonymous variants polymorphic in humans. Now, if we saw a lot of synonymous variants between human and chimp, that might say to us, and we also saw a lot of non-synonymous variants, then we think, well, that's not too surprising. It just has a high mutation rate, and that's why we see that. But in fact, what we saw, nothing particularly interesting in terms of the variation within human populations. But what was interesting was a very strong signature of adaptation in the human lineage after divergence from chimps. We find 13 fixed non-synonymous variants that are in assorted regions of structural domains of the protein. And I find this intriguing. There's clearly something very unique about this protein that is very important and adaptive specifically in humans that would be worthy of follow-up studies. And then lastly, Anurag Verma and Giorgio Sarugo looked at exome sequence data from the Penn Medicine Biobank. They looked at 15,000 individuals who were roughly divided equally between people who self-identify as African American or self-identify as European American. And one of the tests that they did is a gene-based association test. So just take all the non-synonymous variants that were found in any particular gene and look for association with phenotypes that are related to COVID. And we see that there's a significant association at ACE2 with in European Americans with hepatic coma and in African Americans with hepatic encephalopathy. At TMPRSS2, we only see significant association in African Americans for respiratory failure. Then he did what's called a phenome-wide association study where he looked at all the SNPs and their association with all the phenotypes in the database. And here he's looking specifically at those regulatory variants that we found. I will mention when he looked at the coding region, he found an association with myocarditis in African Americans, and in the regulatory regions, an association with type 2 diabetes and hypertension. At TMPRSS2, found an association with abnormal glucose and glucocorticoid deficiency in African Americans. So variation at these genes could possibly shed light on some of the clinical phenotypes that we see associated with COVID. So I just want to end by telling you a bit about the Penn Center for Global Genomics and Health Equity, which we established roughly a year and a half ago. I'm the director of the center, and this is Dorothy Hammond, who's the assistant director, and Amina Alamin, who is executive assistant. And the mission of the center is to promote health equity through global genomics research, education, outreach, policy, and practice. We want to facilitate and promote research focused on genetic and environmental risk factors that influence health disparities in the U.S. and across the globe through interdisciplinary collaborations, community partnerships, and education. We want to translate those findings into public policy and clinical practice to promote health equity. And we want to promote inclusion of ethnically diverse individuals in genetics research and understanding of ethical, social, and political challenges. So I strongly encourage anybody who's listening to this, to studying health disparities or you want to study them with a genetic focus, please contact us, and we'd love to have you join as a member of the center. So when we started the center, and it was roughly like January 2020, and then we had the COVID outbreak. And this is the most striking, obvious health disparity that needs to be addressed. And as we can see, this is a map that I pulled down from the Department of Health just yesterday, showing the percent positivity in Philadelphia. This is up until like last week. And we could see it's particularly high in North Philly and West Philly and Southwest Philly. And we know that there are major social determinants that are playing a role in why some people are getting sick and why some people aren't, as shown here to the right. But what we did is we applied for a grant from the Pennsylvania Department of Health. We've just been told that looks like it's going to be funded. And I should say this is a large collaborative effort with many people at Penn, including Dan Rader and many others. And we're using a community-based participatory, and I should also say we also have some funding from a donor who also has contributed to this. We're using a participatory research approach to study social, environmental, and biological, including genetic, viral, and immunologic factors that affect the risk of infection and severity of COVID, as well as response to vaccination in the African immigrant and African American communities in Philadelphia. We think this is important to assess the true prevalence and measure risk factors that are contributing to this burden and to make population-specific recommendations for public health policy and implementation. Additionally, we hope to determine whether there are differences in the human host, the SARS-CoV-2 genome, and immune response that impacts severity of infection and response to vaccination. And addressing these questions will result in optimization of treatment and prevention strategies. But I'm particularly excited by the fact that we have funding to establish an undergraduate training program with students from Lincoln University. They're going to be called the Lincoln Penn Health Equity Scholars. We hope to have our first cohort next summer, but we also have funding for them so that during the school year they can get prepared so that we can maximize success when they come in the summer. Anybody interested in hosting these students, please let me know. And with that, I'd just like to thank the many collaborators and funding agencies, and I'm happy to answer any questions you may have.
Thank you, Sarah. I don't see any questions, amazingly, in the question tab here. Does anybody have a question before Sarah goes? Okay, you've used up all the time anyway, so
Okay.
thanks a lot,